thing that's always driven me is to try to find out what is the question that really needs to be addressed.
Between the cellular lab and the genomics lab, digital pathology, all of those things, what do you see as the potential for what we're building?
Well, the potential is that you can start to assemble pieces based on what the question is that needs to be answered. And you can approach it in a novel way, not necessarily replicating what someone else has. Part of the challenge with COVID, I think, is people were actually seeing the scientific process as it occurred, which is why the information kept changing.
Where do you think we are right now with AI, not just in health, but as a society?
AI can be very useful, but it also can be very much abused. That's the concern I have. Technology is the instantiation of a solution to yesterday's problem and doesn't know what today's problem is.
Michael Liebman, welcome to the podcast.
Thank you.
Really excited to have you here today, and for the listeners, go over Michael's bio. Michael's, I consider, a good friend and mentor. He is one of the best thinkers I've ever met, and you've taught me how to think a lot over the last 6 years. It's been eye-opening, and I try to take the lessons. And every time I talk to you, I learn something about a way to think or how to grapple a question, and it's been eye-opening. And we've been talking about critical thinking this week, for instance, and the importance of it. But for the listeners, Michael is on our scientific advisory board. He was the chief scientific officer for United Cancer Centers when we were launching that. Unfortunately, COVID happened and kind of stopped that launch. But he has a PhD in theoretical chemistry and protein crystallography. I still don't know what protein crystallography is. Let's go over the bio here. He was he is the managing director of IPQ Analytics and Strategic Medicine. He was the executive director of the Windber Research Institute. He's an adjunct professor of pharmacology and physiology at Drexel. He was the director of computational biology and bioinformatics for the University of Pennsylvania Cancer Center. He served as the global head of computational genomics for Roche Pharmaceuticals and director of bioinformatics and pharmacogenomics for Wyeth Pharmaceuticals. Michael, welcome to the podcast.
Thank you.
I could have kept talking. Here. I could have just kept going on the bio because it's even longer. But thanks for coming to Nashville.
Well, I appreciate the opportunity, and it's always good to see you.
Yeah, absolutely. For the people listening, why don't you give a brief overview of your background, how you got into science, and kind of how your career unfolded over the last, 50 years.
Well, yeah, it's been that long. So my background's a bit unique because I've gone back and forth between industry and academia several times. I started out with an academic career like most people at Institute for Cancer Research, and then went on to Mount Sinai as a faculty member. But what I've always been interested in is the issue of disease and helping understand disease to improve patient outcomes. And while I have formal training in chemistry, I've never been in a chemistry department, but I've been able to take advantage of a lot of different opportunities to learn pieces from them, learn different perspectives from them, and try to assemble that, sew that together to address problems that I think are complex problems. They're hard problems, but I think we really need to start to look at hard problems because they're not going to get easier on their own. And some of the issues I see today are that we're tending to try to use technology to address problems without always understanding what the problems are. Mm-hmm. And the one thing that's always driven me is to try to find out what is the question that really needs to be addressed. So even when I moved into industry, one of my jobs was to go back to academics and find new technologies and so on. And I explained uniquely, I went to Amoco, the oil company, when they were in biotechnology, that showing up on an academic campus, coming from industry, people would want to know how many bags of money I was carrying. And that wasn't the goal. The goal was to really look at the science and form relationships. So what I uniquely carved out that I recommend to a lot of people is that I could also have academic appointments when I was in industry. And that way we collaborated, we looked like scientists, which we were, but we also could publish periodically, and that enabled me to go back and forth much more readily than if I had only gone into industry and not have that set of academic credentials, so to speak.
Yeah. So for the people listening, there's a few different parts of science, but you have industry and then you have the academic world, and they are not the same by any means.
No.
So that is a unique position. Talk about really the differences between academia in science and industry in science.
Well, I'll give you my impressions coming into academics, going to industry, and then coming back. So when I was first in academics, I was in academics for about 10 years. One of the challenges I found at the time, which has changed somewhat, was the lack of interdisciplinary activities. There was a tendency to stay within your lane, in your department, in your field. And I was more interested in solving problems, which meant that would require you to bring in different perspectives. And collaborations were just a natural way to do that. Industry has a different approach. Industry, as they say, there is no I in team. And industry functions usually based on teams. And so part of the opportunity going to industry was to explore how we could really push some of the collaborative efforts forward. The challenge, of course, is in academia, resources are more limited. In industry, the tendency is to have more resources, but the goals tend to be more limited. And so some of the freedom that you would like to have, you don't typically have. I'm not going to say you never have, but typically you don't have. And so as I went through several industrial or commercial opportunities, I to be honest with you, felt I was being opportunistic. If we wanted to solve a problem and I got to a point where I didn't think we could do it, then it was time to look for another place to try to do it. And eventually run out of places and run out of opportunities because, again, I was interested more on understanding the disease than necessarily developing a drug without understanding the disease. And I went back to academics thinking that had evolved. And it didn't exactly evolve the way I had hoped it had. Everyone wanted to start their own company. And, the biotech revolution was taking place, the .com revolution. So everyone thought they could be an entrepreneur as well.
Right.
And so there's a very different perspective. One side tends to have more resource, but much more goal-directed. The other side has more freedom, or the perception of more freedom, but is much more resource-limited.
Yeah, and a lot of people don't know, but for instance, NIH funds a lot of the research at the in academia in the United States, and they have about a $45 billion a year budget overall, give or take. And kind of the model, at least as I see it, and you can correct me here, but a lot of people go work for NIH for 25 years or so. They leave NIH to go work for pharma, many times on some of the similar technology they were working on in NIH.
Right.
NIH then funds the pharma companies, that they're working at, including academia as well. And it's kind of like a cycle. So while academia is different in many ways than pharma, and different than NIH, they all really are connected together because you need them all to develop these drugs.
I mean, in principle, you've got an ecosystem. Mm-hmm. And in principle, academia should have more freedom to explore things. The way the funding has evolved over the years, though, you'll see that it has taken on a bit more of a business orientation, mm-hmm. driven by, what's fundable, what's popular, especially as technology comes along. Mm-hmm. What are the new technologies we would like to apply? And so that has tended to have an impact on the kinds of research that gets done. And the people that are going in looking at new careers, mm-hmm. coming out of their programs, graduate programs, postdoctoral programs, they have to understand that is what academia has started to evolve towards. Mm-hmm. In industry, a lot of the kinds of things that are NIH funding, which you mentioned, it's not the same kind of funding as you have in academia. So you don't have sort of a basic research effort. Mm-hmm. You have more cooperative research agreements, which are contracts that NIH sees that industry can deliver on, mm-hmm. because they're used to delivering on contracts, mm-hmm. that academia is not as attuned to or trained to be able to deliver.
I see.
And, when I was in industry, we would see this as well. A lot of industry works very closely with academia. Mm-hmm. It's a challenge. Mm-hmm. Industry doesn't always, they understand they're not always going to get exactly what they would like, mm-hmm. out of that relationship. Mm-hmm. And academia frequently looks at it as a source of funding, mm-hmm. But not necessarily with the same guidelines and constraints that industry would like them to perceive, mm-hmm. or understand.
Yeah, but NIH can fund both, though, too, right? So it's like,
but in different mechanisms. Not the same mechanisms.
Has that cut down the research cost of the pharma over the years? Because I've seen debates about.
It's not a major input. I mean, you may have it in a very directed program, like a vaccine program, because there's a need for a vaccine program. But in terms of overall research and development costs, pharma really puts the majority of that funding in.
I see.
You can look at the numbers from the pharma organization.
Mm-hmm. And you're also director of translational medicine for the Pharma Foundation too, right?
I was until just recently, both for translational medicine and for informatics.
Talk about translational medicine a little bit. You're also an editor for the Journal of Translational Medicine.
Right. So
what is it?
Yeah. Pharma, several years ago, wanted to set up translational medicine as a program. Pharma has a philanthropic arm called the Pharma Foundation, and the goal of that is to promote education. Mm-hmm. It's not tied directly to the Pharma Institute, mm-hmm. Pharma industry. Mm-hmm. So it's not to build a feeder farm, so to mm-hmm. speak, for the pharma industry, but to sponsor research that pharma sees as a need long term. Mm-hmm. So informatics had been involved for a long time. I started when it used to be called bioinformatics, but we expanded it to include clinical informatics and so on. And there was an interest in creating translational medicine. Actually, translational medicine and therapeutics was the original title. And so we put together, or Pharma put together, their board, a process involving McKinsey doing a survey, and then brought together a very large group representing government, industry, academia, to discuss what a program would look like. And having been in academia and in industry, I had raised the concern. They asked me if I would share it to get it started. And I said I would, but we needed to, I wanted to have certain freedom in doing that. And the freedom was, let's not take what at that point was the conventional view of translational medicine, which was, let's take research out of the laboratory and move it into the clinic. And the reason was that while I was in academia, I saw incredibly great science being done. The concern I had was I didn't see that it necessarily was solving clinical problems. And so the concept of that translation was not really bridging the valley, so to speak. And yet the academics felt if they were doing really good research, it should convert to clinical utility. And the clinicians and basic scientists don't exactly speak the same language. No. They have different missions.
Yep.
So when we started this program, I had people from the Gates Foundation, from different NIH or government groups, academics and industry. And what we decided to do was say, if we're going to do translational medicine, and the goal is for research in the academic environment to be able to move to the clinic, we have to start understanding first what the clinical problem is. And therefore, we're going to start and take not a bench to bedside model, but a bedside to bench to bedside model.
Interesting.
So if we take the problems out of the clinic, bring them to the bench to work on, then we know that the solutions will have an immediate application.
Yep.
And we've closed that gap.
That's interesting. So, for people listening, translational medicine in general would be considered bench to bedside. That's like the standard definition. And basically translating the findings in the lab on the bench to the bedside of the patient through the clinician.
Right.
And, one of the interesting things that I've found running the hospital and being so involved is, I think from the outside, most people think that doctors are scientists.
Right.
And scientists, and somehow, and that really was the case, 50 or so years ago. It was more of the case where clinicians would make discoveries and they would notice things and they'd, really expand upon that. But what you're saying is actually a little different. I haven't ever thought about that way, bench to bedside, back to bench. And, I like hearing that because that's kind of the model that we have set up in Mexico, where we have, our 6th floor cellular lab, we have our clinical lab, genomics lab, digital pathology, and the doctors and scientists work area is the same. So they're working collaboratively to do that.
There's a couple of things to point out, I think. One is the majority of the way medicine's practiced right now, physicians have to be operational. They're not given enough time because of the way it works to really devote as much time as they might like to devote to understanding the patient and their condition. And the patient is putting pressure on the physician to come up with a diagnosis and a treatment. And actually, the internet doesn't necessarily help because they're coming in armed with all kinds of information and misinformation. And the doctor frequently has to sort through that as well. So it's not a perfect match. One of the things we've tried to stress is let the doctor be operational, but let's find out what is the strategic side, not the operational side, that they really could benefit from having more effort to be involved in. And, a common thing would be, an easy thing to understand, the doctor has to come up with a diagnosis, okay? The diagnosis gets tied to treatment, it gets tied to reimbursement, hopefully to improve patient outcome. But they don't have time to really study the underlying pinnings of what the diagnosis actually means. And so we spend a lot of time exploring how do you make a diagnosis? How accurate is that diagnosis? Does the disease have to be stratified? Because maybe there's 5 different subtypes in that diagnosis.
Yep.
And the doctor doesn't have the tools necessarily to apply it that way unless they have the experience already and they've seen it. So we try to take that on. Now, why that's important is when I left academics, we were trying to do a lot of that, but it wasn't sort of the current model. And there were a lot of things we were trying to do because this was very early, and it gets to the model that you have in Mexico. We're trying to say, instead of trying to take all the databases and put them together, the way people even now try to do, let's change this and make it a patient-centric model. Because in essence, the patient owns everything. They own the tissue, they own the disease, they own their lifestyle, their environmental exposure, their culture, their social determinants. And let's make it patient-centric, because that really is what is all about. And let's use that as a computational model as well, because now we can make the model of all of the data represent the patient. And we can ask questions about that.
Yes.
That wasn't the way things were going at the time, which was around 2004 in the academic medical community. And so that's when I left to run the Windber Center, which was a Department of Defense breast cancer center, working with Walter Reed. Because whereas where I was in academics, I kept being told, well, we can't do that. That's not the way we do it. When I presented it into the DOD, it was, well, if it makes sense, we'll do it. And we don't have those barriers. We don't have those structures.
What was that like? Like when you were able to do that and go from all the barriers to being open, what did you accomplish?
We could ask questions that we couldn't start to ask before. We would see things, relationships among data, among patient characteristics. We would see, the most important thing is we could start to better see where there were gaps and conflicts. Because the thing about science is it's constantly evolving. And it's never going to be perfect. You're never going to understand everything. But if you can identify where there are gaps and conflicts, you have a better idea of where to prioritize your research and your investment. Which ones are important? Where will you get the return on investment, so to speak? And changing the model gives you more freedom to do that, because we weren't going to have to align with what was an NIH review panel going to want to do when everyone on the panel did the same thing.
Right.
And you're trying to change something.
Yeah, give an example, working for the DoD, a change that you were able to make that made a positive difference.
Well, the data model, we went from the concept of an EHR, electronic health record, where you would put all your data in, to a personal health record. And that started to then, so the idea would be a patient who would come in to a doctor's office and you would ask, do you smoke? Do you drink? How much do you smoke? How much do you drink? Because I'm trying to assess your risk for breast cancer. But the reality is that patient actually undergoes developmental change from the time they're a fetus during pregnancy all the way through the rest of their life. And the breast is actually a perfect example of that. There's 10 different stages of breast development just during gestation. And exposure to these factors like smoking, or which aren't from the fetus, of course, but maybe the mother and the environment, they're going to have different effects at different points in time of that development. So risk is not something that is as convenient to model as asking, do you smoke and maybe you should stop smoking. It's maybe when you were smoking at the time of menarche, that was when risk actually developed. But we couldn't ask those questions the same way, but we were free to do that kind of modeling and expand upon that with the DoD experience.
Yeah, so that was, you said 2004-ish.
Yeah.
I guess the Human Genome Project was already underway. That started, in the early 90s, I believe it was, late 80s, early 90s?
Yeah, actually, when I was with Amico, it was before the Human Genome Project started, and we were working with Carl Woese at the University of Illinois on the Prokaryote Genome Project. And the idea was that would be a model for the Human Genome Project. So Amico was involved in that. And then Amico, of course, because it was an energy company, was involved with the DOE, the Department of Energy, and the initiation of the Human Genome Project.
Yeah, I mean, it was a heck of an accomplishment overall. I mean, if you look at it, what was it like kind of living through that? I mean, I'm sure, you had some scientists that were like, this is going to immediately make a big difference. I say this because I think about where we're at with AI right now. And, maybe
it's interesting because I remember I was at a meeting in Waterville Valley, which I remembered being one of the very first meetings talking about this. And Wally Gilbert got up and said, I'm going to sequence my genome. Okay. And you can see how far we've come since then, because not everybody believed it at the time.
Interesting.
So we've come a long way. And you refer to AI, we still have the challenge that we can generate a lot more data than we can extract both information, which is clean data where we have context, and definitely much slower to create knowledge or clinical utility out of that. And so that's really the challenge. We keep developing new and new technology to give us more and more windows, so to speak, on the problem, but they're not necessarily all developed because we know what the question is to ask.
Right.
They come about because we have the technology that can make the measurement.
Yeah.
So it's a bit of looking for your keys under the lamppost. But now with big data, we have a lot of lampposts, a lot of people looking for their keys, and they may still not find them.
Right. Well, you've been involved in understanding AI, and you work with quantum computing as well, for a long time. So, and it's interesting as this AI, the last year it's gotten popular with ChatGPT, and but you've been working with this type of stuff for a long time and have understood its limitations.
Well, we started working with AI back in the 80s when I was with Amico. And actually we brought it into Amico at a certain level, and we were called into other parts of the organization. We were obviously interested in applying it in biology and healthcare, but we were called into the refinery. We were called in to work with the credit card division because they wanted to see if AI would solve some of their problems. But one of the things we found was we could take problems that seemed to have been solved using conventional statistics, and we could apply the AI, in that case it was neural networks, and find more complex relationships that hadn't been seen. Because the statistics would give you not that the neural nets were not a form of statistical analysis, because they were still correlative, but they would allow you to look for secondary and tertiary interactions that you wouldn't always find through the other means. So, but at the same time, that still, and to this day, is still a challenge, because we're still interested not in correlation, we're interested in causality. Because if we really want to change medicine and improve healthcare, we have to understand what is the cause of the condition. How early can we detect the condition or someone at risk for the condition if we want to make and have an impact, not are these symptoms related to the condition.
Yeah, and one of the things that you've explained to me well is the AI is only as good as the data that's being inputted. So if the data is all wrong, then you're going to get the wrong answer ultimately.
Yeah, there's a lot of emphasis on big data. And the challenge is not all data is created equal. There seems to be a bit of a difference between data scientists and data engineers. I'm working right now with a data scientist, and her training didn't expose her to really looking at what the data represented as much as how do you process the data and use the methodologies. And that's one of the reasons she has started working with me, because data that has the same label in different databases doesn't mean it was collected the same way or means the same thing. And as a result, you have a problem, because when you put that together to create big data, the data is very heterogeneous. So I'll give you an example, and actually as it translates to real life. So I always use this example. The gold standard for understanding kidney function is glomerular filtration rate, which involves the processing of fluids by the kidney coming in and going out. The most correct way to measure that is to put the patient in the hospital, control the fluids, and have accurate measurements. But that's complicated and complex, and most physicians don't want to put the patient in the hospital. So what they've done is they've created what they call estimated glomerular filtration rate, eGFR, and that's in a lot of patient records, and it's computed using some other laboratory measurements, creatinine and et cetera. But what's not necessarily acknowledged is the fact that there's at least 5 different equations to compute eGFR. They were developed for different reasons, so they're not directly equivalent. They don't give you exactly the same number, and 2 of them have a race factor involved, which has been disproven. And so if you just take different databases and put the eGFRs together, you don't know where it comes from, and it's not annotated. And so you're putting data together that may not have the same quality or the same basis. And to show you how that really has an effect, I was having a conversation with a transplant surgeon who was acknowledging this issue at HIMSS. And 2 days later, CNN had an article, a report of an African-American woman who had been on the transplant list for 5 years, but was not given a transplant because they used the wrong equation to compute her risk. And once they switched away from one of these race-based equations, she suddenly was elevated on the list and got a transplant.
Gotcha.
So she actually was able to be treated appropriately, but had a 5-year delay in that treatment. So these things really do matter.
Well, yeah, and with the AI, I mean, there's a lot of applications, but one of the things that you've taught me is, a lot of this is based off of people's medical records, and the medical records are based off of insurance codes in many ways.
Well, some aspects, yeah.
Some aspects. Yeah. And so you get these data points that they don't give you really a clear answer of what's going on. And let's say that those data points give the specific diagnosis, but what it might mean under one insurance claim is different than on another as well. And so they're getting bad data that then, if you're using the AI for that, there's no way it can actually know because it's given the wrong information.
Well, so just to clarify, the clinical data per se has a kind of problem, could have a problem like I just outlined.
Right.
But tends to be numerical and therefore a bit more reliable. Sometimes, though, the data that's recorded is based on setting a threshold that something's positive or negative. And then it depends on who's setting the threshold, because different guidelines may set the threshold differently. And that's something that you need to take into consideration. The challenge, which is primarily in the U.S., is that the majority of the data that's available for analysis is claims data. And as you said, that's tied to reimbursement. It's not necessarily tied to understanding the disease itself. And so I always find it interesting because my genetics groups or friends who are always looking for genes associated with a diagnosis and are using a diagnosis code, an ICD-10 code as an example, in the U.S., they are using a code that when you talk to the clinicians, they'll tell you, well, we had to put that down because we needed it for reimbursement.
Yep.
It had to justify the tests we wanted to do or the treatment we wanted to use. It's not necessarily representative of the disease. And so that tends to obscure that. Now, outside of the U.S., where you have nationalized health care, the diagnosis code could be much more reliable than it is when you're using the claims data that's based off of a reimbursement model.
I see. It'd be interesting to do a study comparing same patient claims data versus their medical records and see what the different outcomes are. Have you ever seen that done?
I haven't seen that. I mean, it's definitely something that would be of value.
Yeah, no, absolutely.
Someone must have done that.
Someone had to have done it. Yeah. If not, we need to figure out how to do it. So let's go back a little bit to translational medicine.
Right. So just to back up to where you were talking before, the same transition going to the DoD and not having as much of the embedded culture in place is what you have the opportunity at CHIPSA to do, because you're not working with an embedded culture and you can focus more on the problem and bring the pieces together necessary to try to address the problem.
Right.
And so that's where a lot of these opportunities can be seeded and started.
Yeah, no, it's exciting because we're kind of getting to build this from the ground up.
Right.
And what most people know us for now is stem cells, you know, the CPI model. But there was CHIPSA well before that, and we're actually in the middle of the name change, Translational Advanced Medicine, TAM Center, Translational Advanced Medical Center, I should say, TAM Center. And we're really going, you know, not dismissing what we did with CHIPSA, because we did a lot of great work, but evolving to where it's all science-based. You know, we were having the discussion yesterday. It's going to be hard for someone to look at what we're doing and say this isn't science. We could have bioethical questions on how that we need to solve, and I'm fine having those conversations. I think about it a lot. I know you think about it a lot as well. Our whole team does. But you know, what do you see as the potential for the lab that we're setting up between the cellular lab and the genomics lab, digital pathology, all of those things? You know, and some of the things we were talking about yesterday, those extra, let's say, 10 steps or 10 other things that we need to figure out in between. What do you see as the potential for what we're building?
Well, the potential is that starting from scratch, as you put it, you can start to assemble pieces based on what the question is that needs to be answered, okay? And you can approach it in a novel way, not necessarily replicating what someone else has. You know, I once had, we were working in a pediatric NICU, and a colleague of mine brought me in because we were working in pediatric ARDS, and there was an infant lying on the bed. The parents were in the room. There were 5 people looking at this instrument. Baby was strapped in, and the instrument had 5 dials, basically, to make it simple. And they were turning the dials and seeing what the response was and turning the dials. And we spent about an hour there. And we walked out. My colleague was the head of the NICU. And he said, so what did you think of what you saw? And I said, well, from my perspective, I just saw a random walk in 5 dimensions for an hour. Because they couldn't tell you if they repeated anything. Okay. And I don't know what the coordination was of all the dial, because I'm not a clinician, of course. I know that there was experience and so on that they were looking for and response. And a month later, I was at a Midwestern medical center, a big medical center. And I happened to be talking to the head of training, MSTP, Medical Science Training Program. And I said to him, humans are really good in 2 dimensions. Number of humans are okay in 3 dimensions. 4 dimensions is very hard. 5 dimensions is impossible, okay? So how do you train your physicians to deal with this? Because it is a 5-dimensional problem. And he said, well, we only really train them on 2 dials at a time. Okay. And so I said to him, so why do you have an instrument with 5 dials? I said, is it because the engineer knew that each dial was worth $25,000 and you put 5 dials on the box and it's now $125,000 instead of $50,000? And he laughed, and I said, or is it because the hospital across the road has the same thing? And he says, because the hospital across the road has the same thing. And people are going to expect 5 dials.
Right, exactly.
Right?
Yeah.
And so part of the challenge from the way we build models is we treat disease as a process, and a chronic disease also has human development under that. And the challenge of that, of having disease as a process and not a state, because we talk about a disease state, is that you have to look at how things change over time. And that is a big challenge, because if a patient comes in with a disease, a presentation of symptoms, it's not ethical to not treat them and just allow them to progress to understand what the disease is. I mean, you may do it for a small amount of time to see if they resolve, but you're not allowing them to run the full course that way. And so we don't know what the real natural course of a disease really looks like, how many dimensions in terms of clinical parameters it involves. And if you intercede that at different points in time, and you're only extracting the data points at that point in time, you may diagnose it differently, because different diseases may cross at different points in time as they evolve. And now what you're trying to do with technology, diagnostics, and biomarkers is you have something that can make a measurement. And that technology and that measurement, you're trying to figure out how to align that with that disease process. And it may be very different at different stages of that disease process. But if you don't know the disease process, it's very hard to do that alignment. And so you're constantly fighting that battle, that challenge of getting that proper alignment to be able to really hone in on what we call, what is the disease vector in high dimensions, how far along that vector a patient is, which is their stage, and then how quickly are they progressing? Because if they're progressing slowly, you manage them differently than if they're progressing rapidly. But if you take those 3 parameters, then what you really have is you have a tensor, not even a vector. And I call it sort of the Heisenberg uncertainty principle of disease, because you can't measure where a patient is on that vector and how rapidly they're progressing at the same time, which gets to the physics part.
Right.
So that's the kind of challenge we have. And I don't know that we'll completely resolve it. A lot of the new digital monitoring, 24-hour monitoring, can help give us a much better profile of these changes over time.
Yeah.
And that's one of the big values of how we should start to use that data.
Yes.
But it's still going to be a very complex marriage of the data with
points.
understanding what a disease process is.
Well, let's talk about that for a minute, because just to give people an idea of something you're working on, with the underserved communities, specifically in pregnancy. What are you working on with that? How are you setting up that model with the earpiece and those type of things?
Yeah, so we have a women's health program, and it starts with preconception. We spend a lot of effort in infant and maternal morbidity and mortality, which is a very big area of health disparities. We go into postpartum and developmental issues, and then we actually go into perimenopause and menopause and postmenopausal disease risk, cardiovascular disease and breast cancer. But looking at the infant and maternal morbidity and mortality, one of the things we focused on are hypertensive disorders of pregnancy, which are very common. The most common one, the most notable one, is preeclampsia, which most commonly results in premature birth. The cost in premature birth and the result of these hypertensive disorders is somewhere in the first year aggregate in the U.S. over $2 billion.
Wow.
And of course, these can have a long-term impact on the infant. So it's not just that they resolve, but they may have developmental issues and so on. So we became interested in the idea that we know with the health disparities, there's a much greater risk for preeclampsia and hypertensive disorders, say in the African American community than the Caucasian community. We also know that hypertension in general is more common in the African American community than Caucasian community. But it's estimated that in the U.S., half of the entire U.S. population has hypertension. Okay. Globally, it's about 30%, but in the U.S., it's about 50%. So one of the things we thought we would start to do is see how early we might identify that a woman is at risk for developing hypertensive disorders, to try to intercede earlier, monitor them earlier, and also recognize that while we talk about preeclampsia, and this gets to the issue I was talking about before, stratifying the condition, it's not one condition. There's a set of parameters, and it's multiple subtypes of preeclampsia that need to be dealt with. And they may have different targets for drugs, the different response, different critical interventions, and so on. So we became interested in trying to piece that together in a model. The goal was, let's see if we can start even preconception and evaluate hypertension there, or risk of hypertension, and see how that progresses. That led us to start to look at how is blood pressure actually being measured? How is hypertension being monitored? And the challenge we realized is that blood pressure actually in the clinic is not even measured according to the guidelines that exist. You can talk to almost any clinician or cardiologist, and they'll tell you, we don't exactly follow the guidelines. We do it the way we do it. And then based on what they do, they look at a chart and assign whether you have hypertension.
Right.
Okay. But the reality is your blood pressure varies over the course of a day, normally by about up to 20%. And most notably at night, there's a dip called the nighttime dip. And that's known to be different in African American population than Caucasian. And there's concerns and studies as to the rate of fall or the rate of rise in the morning, that people are at greater risk for heart attacks with that rise in blood pressure in the morning. But we are taking one measurement typically to represent that entire transition. And so what we started to do is look for ways to actually monitor that entire transition and parameterize it so that we could take features of it, how rapidly are changes occurring, and then see how those relate or may be predictive of things like preeclampsia, as opposed to that single measurement. And if that's so, can we then look to make a better decision about how to treat those individuals? Because what's the underlying pathophysiology that causes that feature? And then let's get the right drug that attacks that problem. Because right now there's about 12 classes of antihypertensives. And choosing one is not necessarily straightforward.
Right.
Physicians will have their favorites and so on. But you're, in a sense, titrating the patient
Yes.
to see what they respond to. And this is trying to change that, personalize it. And it may change when you get dosed, and it may change the order in which you try medications.
Right. I think there's a few things. There's a few things to unpack. Specifically, the way we measure blood pressure is not really. It's standardized, but it's not standardized. People don't do it the same way. The other day, I was getting an IV, and they were going to take my blood pressure, and I was laying back in the chair. I was like, no, I got to sit up and get my feet on the ground. And then the other nurse was like, oh, yeah, you're supposed to do that. I'm like, well, that's a great example. I thought about you, that they were going to do it the wrong way to where you don't get an accurate view of your blood pressure. The other thing is your idea is you've got a little earpiece.
We're working with a company that developed an earpiece.
Yeah,
we don't have the earpiece.
Yeah,
yet.
sorry. Yeah, you're working with a company, but for your project is to have that earpiece that the patient wears, and it monitors it accurately throughout the day.
Right.
So we understand the answer to that question a lot quicker.
Yeah. Right now what happens is a patient could be fitted with what they call an ambulatory blood pressure monitor, which essentially is a cuff that every 30 minutes blows up just the way your cuff normally does. The challenge, though, especially if you're looking at it overnight, is it interrupts your sleep.
Yeah.
And that's going to cause your blood pressure to change as a result of the intervention to measure your blood pressure. And even during the day, if you know it's about to trigger, you're going to have a response, an anticipation. So while it's much better than a single measurement, it's still not a non-obtrusive way of monitoring. And what this potential opportunity is to look at a recording that's non-invasive and doesn't have that same, it's passive, completely passive.
Yeah. And, what's interesting to me, that's just one application to the model that you created. And to give people some context, 2019, we were launching United Cancer Centers, or we're raising money for United Cancer Centers in the United States. You had a model that, like I say, we weren't ready for yet because I didn't exactly understand it. It takes a minute, I'm not a trained scientist like you, but we were very much we were patient focused at UCC. We were going to be the first institution to utilize the right to try legislation. And you wanted to take it a step further where we had our own genomics labs. We were doing our own pathology. I think either you or VJ brought on the digital pathology. But the idea was to get as much information as we possibly could from the patient's tumor and then deliver precision medicine to the best of our ability and build out the labs with UCC. Now, at the time, I knew a little bit about genomics, like just very, very basic, but hadn't really dove in. And I'm like, oh gosh, I don't know how we'd put all that together. But as COVID happened and that project ended up not happening, it's like, there's an opportunity to do that, in Mexico, and even be able to have more freedom to do more.
Right.
Like my question with the solid tumor genomics lab is, how do we build the best solid tumor genomics lab on the planet, at least for the patient specifically? There's other places that'll have our capabilities, but is it going to be centered towards what the information the patient needs, either now or in the future? Right now, there's going to be discoveries on the tumor that we can't really take action on, but in a year, 2, 5 years from now, there's that possibility. So I like to say that a lot of the idea for what we're doing now came directly from you. This was part of your vision for what we were doing, and I say that because the earpiece is another. I say the earpiece, the project that you have with the earpiece and the pregnant women is another part of that, because the model that we're creating in Mexico, we're going to bring to Nashville. We have the potential to ask questions in a completely different way and focus on these underserved communities to make a huge impact. And you can do that without a lot of money. I mean, it doesn't take a ton of money to, once you understand how to build the labs and run the tests and what tests to run and how many samples you need before you can run the machine at a cost-effective way, there's ways that for a small amount of money, you can get a lot of meaningful information to patients on the cancer side, but also just on the overall life side. And kind of elaborate on that, because I don't think people realize maybe that's what we're talking about in the whole. Like, this is an ecosystem that you're putting together based on how to solve complex diseases and how to really make the world a better place, or a healthier place for everyone.
Yeah, and I guess there's sort of two examples for that. One is, as an example, there's a lot of emphasis right now on social determinants of health. And they're important. There's no question about that. But I think a challenge that we haven't necessarily adopted or acknowledged is, to a certain extent, we're looking from the outside in and trying to make an assessment of where certain characteristics, features should be, could be and should be. Is it an educational problem? Is it an access problem? Is it a smoking problem? Things of that nature. We also need to expand upon that, which is something we've been doing, and say, that's from the outside in. You need to understand it from the inside out. Because if you really want to be patient-centric, and especially if you're going to be dealing with multiple cultures, they don't look at the world the same way you do.
Right.
And you need to understand how they look at the world, because your priorities may not match their priorities. And as much as you can explain it, they may not accept it and act on it.
Sure.
And so it needs to be an interface between the two. And then even with several cultures, at least, you need to understand the concept of trust. Are they going to even trust what you're telling them? Because simple examples in trust are, do they have confidence that you know what you're talking about? And do they have confidence that you're looking out for them, not for some other purpose?
Yeah, absolutely.
And all of these things come into play that make the problem more complex than even just looking at the social determinants. But that's what the real world is about, and that's what a real patient is about.
Absolutely.
And so we need to keep those things in mind. On the other hand, again, with the technology, as I said before, we take advantage of the technology we can develop and then try to use it to look at disease. I'll give the example I'll use is when I was at Vices, we were working on the HER2 new test using fluorescent in situ hybridization, or FISH. That measured gene copy numbers. That was different from the immunohistochemistry test, IHC, which looks at an antibody response to a protein. And yet both eventually were approved for use for looking at what they call HER2-neu positive breast cancer patients, which are about 20% of the patients. Mm-hmm. The challenge was they don't give you the same answer necessarily because they're not looking at the same thing. Mm-hmm. And we have to understand that biology is complicated. Mm-hmm. So going from the copy number of the gene, which ties to the chromosome, to the protein, there's multiple steps and transformations.
Yeah.
And it may be that those transformations are the critical thing, and we're only looking at the endpoints. Mm-hmm. And by not looking at the part in the middle, we're missing what really is important. And the suggestion that that could be a reality is we're no longer looking at HER2-neu positive and HER2-neu negative. We're also looking at HER2-neu low. Mm-hmm. So we've started to increase the classifications.
Yes.
And that shows you at least that science is continuing to evolve. Mm-hmm. But it evolves very slowly because the tendency is to try to come up with classifications and put things into boxes.
Yes.
Whereas the real world doesn't really like that.
Yeah, and a couple of things. I want to actually go back from the copy number to the protein.
Yeah.
And go through that system. But, what you're talking about with HER2, I feel like I have a good amount of experience with. I've seen it in action, right? And I shared with you a case. We had a patient, I'll just share with the listeners, had a patient who metastatic cancer. It was a stomach cancer, didn't know exactly which type of stomach cancer. They were at a specific hospital. A friend contacted me. She hadn't eaten in weeks. She was emaciated. He said, "Hey, can you help her?" I said, let me see if I'll give it to our doctors and see what they think. Got back to him pretty quick, and it's like, she might pass in the hospital within 7 to 10 days. So we've got to be, you got to know that, set that expectation. But we'll give it a try. Well, she gets in, and the institution that she was at is a great institution. It's like 1,000 pages of medical records or so. It's like and we started digging in and noticed that a genomics test had been ordered, on the tumor, and it had not been put in the system. And so we called the next day, and they sent it right away. It had an actionable marker, HER2, mm-hmm. and we'll get into the HER2, what you're talking about, low or regular. But within a week, she was eating. Within 2 months, her cancer was gone, not cured, but you couldn't see it in images, couldn't tell it through blood work. And it turned out she had a specific cancer that's what's called CDH1 positive. And it's a stomach cancer that you have about a 75% chance of getting this stomach cancer if you have the CDH1. And they recommend, if you have it, that you get your stomach removed. Mm-hmm. And which is it's a big deal to do. But she got it right at the age that most people get it at, and lived a couple years. But that's one specifically where we had a target. The HER2 worked well,
right?
And there was a good outcome. What you're talking about, and this is actually the first I've heard this, was HER2 low, right? Which is different than HER2. And 40% of patients that receive HER2 treatment that are HER2 positive respond. The other 60% don't respond. But they might be looking at the wrong marker. They might be looking at HER2 when it's HER2 low. And if we would test for HER2 low, possibly.
Well, HER2 low means normally what they do is they have a threshold. Mm-hmm. And that means how many cells light up with HER2. That threshold, if you're below that, you are negative. If you're above that, you're positive. HER2 low means that that threshold is now a gray zone. So it's not a different marker.
Right. Well, it's a different amount
of HER2.
Yes.
Yeah.
So a different amount of HER2.
Right.
But if they have that different amount, could the response be, let's say, I'm going to throw a number out, 100% or whatever, versus 40%?
Right. So since HER2 positive is usually about 20%, the issue is, and as you noted, only about 40% of the patients actually respond to that. The issue is, from a systems modeling perspective, how do you identify who it's not going to work for and not give it to them so that they can have a treatment that might work for them rather than have a delayed encounter with a drug that isn't going to work. Their disease continues to progress, and the HER2 or whatever the drug is may actually alter other aspects to make it more difficult when you decide that it's not working.
It could be changing their biology.
It could be changing the biology. And that's something that has to be considered from a systems point of view, not just who does it work for, but who should not get the drug.
Right. And so, that's just one example of, blood pressure. How they take blood pressure is another example of ways that, the system, this doesn't make people bad for doing it this way. This is the system, but how the system has been created to do things in a certain way. And we know there's a better way.
Right.
I mean, there's things that are just blatantly obvious in medicine and science. You're like, if you did it this way, we would actually get a better answer to the question that we're asking, to the problem.
A lot of times when I speak with clinicians, because I spend most of my time talking to the clinicians, when I present some of these, what I consider challenges, they go, yeah, it's a really hard problem.
Yeah.
But it's not one that I can solve.
Right.
And I have to do what I can do. And that is an acceptable and understandable answer. But that gives us the idea of where we need to try to contribute to take away that hard problem and reduce the complexity.
One of the things that I enjoy about working with you is watching you with the clinicians. Dr. Escobedo, our medical director, has a lot of respect for you. He's like, he said, Michael asked the right questions. And, I think Dr. Escobedo shared with you, he's been noting some of the questions that you're asking on how we do exams his entire medical career, right?
Yeah.
I mean, that's so the blood pressure is one. There's another one that you brought up in the past. Is it with breast cancer? Gosh, that's a common way that they're when they started their period for the first time, right? Isn't that like a determinant of specific? Or that may be something else.
Well, we are looking at menopause, and what we're looking at, we're actually initiating a survey because gynecologists are not well trained in menopause. And you can ask any woman age probably 35 or older, they don't get the information they really need. Many of them don't know if they're in perimenopause, let alone menopause, and that can be 10 years long. So one of the things we're doing is we're initiating a survey in the US and in Canada to collect initial data to try to see how women go through perimenopause and menopause under common pathways, so that with a limited number of pathways, you can better understand what to expect. So as opposed to what a lot of women do is ask their friends, have you had hot flashes and what did you do? As we were talking about before, hot flashes may be at a different point in that transition for one woman than they are for another woman. And what's important that you may really need to know is, are those hot flashes associated with brain fog? Are they associated with other symptoms? or were other symptoms present before the hot flashes? Because that may change how you respond or manage that. So we actually are initiating a survey to collect initial data to look at that. And the kinds of questions that we change, to give you an example, because we're starting with understanding when menarche, from menarche all the way through to perimenopause. The kinds of questions we're looking at, they're frequently simple statistical questions. This is where the AI is applied. How many pregnancies did you have? But what's really more important than the number of pregnancies you had is how long was the interval between the pregnancies. Because obviously not speaking from personal experience, but a woman who's gone through a pregnancy will recognize that she's undergone physiologic change. She's also undergone changes she's not aware of that may have to recycle and reset before a second pregnancy comes in or is initiated. And those gaps or those periods of time could be very critical in understanding how and what symptoms or how she will progress eventually to menopause. So that's where looking at these problems, but going to a much higher degree of resolution, we think can be helpful to better understand the real physiology.
Yeah, no, absolutely. And as we were talking about yesterday, from a modeling standpoint of, for instance, let's say a solid tumor cancer, I mentioned methylation and you kind of chuckled. You're like, well, it could be methylation or it could be about 10 other things, as in the processes in between. But, going from, the genomics all the way, down to the clinical lab or even the digital pathology, to give people an idea, like what we're talking about doing to start with is whole exome sequencing on all tumors. We're also going to be doing a 1021 gene panel as well, which is a bit redundant, but this it's a good double check. As you know, how the sequencers work, as you mentioned earlier, the 30x means it does the test 30 times basically to see if there's errors. We're also doing cytokine testing. We're also doing digital pathology, optical genome mapping, combining the clinical lab data as well. But it could be the in-between those things, how the process kind of flows through from the genomics to the protein that the answer is. And while we'll get a lot of information to start, really good information, more information that's useful than elsewhere, we're not able to exactly solve this huge complex problem until we set up a lot of other tests in between.
Biology is about processes. Just like I said, disease is about process. Development is a process. Going from what information you have at the genome level to how that eventually translates into risk for disease or actual disease involves a number of different processes. Some may occur at a DNA level, some may at an RNA level, some may at methylation, some may be at the protein level. And you don't know if it's one or more than one that is actually involved. So what we're talking about is trying to lay out at conceptual level what are the different steps, and then map the technologies that are available to see which steps and which parts of that process we're actually collecting data on, and try to segment it to get an idea where a certain kind of cancer, maybe we're looking at the wrong segment because that's where the technology is. It's not that the same technology is necessarily going to be uniformly applicable across every cancer type or every disease type. And so that's one of the things we're trying to lay out, and then see if there may be a way to prioritize the next technology we try to develop, the next set of measurements, or combining existing methodologies to give us other answers
Right.
that we haven't currently pulled out yet.
Right, exactly. It's interesting looking at this, not as a scientist, for me, like coming in saying, okay, well, it's the first difficult part's figuring out all the different equipment that you need. Like from a basic level, it's like, okay, what equipment do you need to do this test? Who do you need to hire to run that equipment because it's advanced? What tests do we want to run? We've got this great lab we're setting up, but I'm still learning constantly. Like, we were talking about yesterday. We got to see if we need to buy a new machine for this, or we might have to look to the outside to get a test developed. Or
it still always should start with what's the question you're trying to answer.
Right.
And if the question is, are we looking for protein markers on the surface of a tumor? That's a different question from asking how early could we have determined that someone was at risk for cancer.
Right.
And so different questions require different parts of that entire process to be the focus.
Right. Or what's driving the cancer right now.
Right.
It's pretty interesting, but we're going to have a lot of capabilities. And I think that this model, it can easily be transferred to the U.S. I mean, we'll be doing it here.
Yeah, it's not unique in any way.
No, it's in many ways common sense. I mean, it's a common sense approach to trying to solve a complex problem.
Yeah. Well, you and I have discussed. It's basically what I call applying critical thinking to a complicated problem.
Right.
It's not. We're trying to take a complicated problem apart, look at the root cause elements of that, and then see which of those we can address or we can't address, and what that is going to mean about biasing the results we get and interpreting those results and then using them in the clinic or using them to develop drugs or other treatments.
Yeah, no, absolutely. And, from what people are used to, let's just say right now in the genomics field that's available to patients, you can do whole genome sequencing. You can do whole exome sequencing, which is the coding regions. The whole genome sequencing obviously is the entire genome. The whole exome is the coding regions of the genome. You could do targeted panels, which are the...
Well, maybe you should explain to the audience. The exome are the parts of the DNA that code for proteins.
Yes.
The proteins actually do the work. Mm-hmm. The exomes represent only a couple of % of the entire genome. Mm-hmm. So when you're doing a genome sequence, only maybe 1 to 2% of that is actually converted into proteins. Mm-hmm. The rest of it, we don't actually understand.
Yes. So that the exome is the part that we somewhat understand. The rest of the genome, while you might not want to sequence the whole thing, I mean, it's a lot of data. And, it's...
But the bottom line is we don't know what we don't know.
And we don't know, yeah.
Right.
But the exome is actionable in some ways. We know something about it. And so, yeah, you go from the whole genome to the exome to the targeted type panels, which are good too, because they're the ones that we know a lot about. We know that maybe they cause X, Y, or Z as well. And so in doing that, the tests that you can get right now, let's say in the U.S., you send your blood work in, you get a whole exome test or a whole genome test. You're going to, or a targeted gene panel test. It's going to take 4 to 6 weeks to get the information back. And then you'll get a variety of reports. And those are available readily right now. The reports, though, it's pretty interesting because, there's a lot of bioethical debates about the information that you get. People have to be aware. The article that you just gave me was a good one. You have to be aware that, okay, you do this whole exome sequence and you get a report that says you're at a high risk for heart disease, all of a sudden you might not be able to get insurance on, on your life insurance. And, should it say necessarily high risk or medium risk, and who makes the determinants of what is high or medium? And I'm just using that as one example. There's a lot of questions that need to go into that you need to be aware of when doing, these tests, and things to consider as well.
Well, it's unfortunately even more complicated.
Yeah.
Because the changes that you see that you're now associating with that risk, as more research gets done, sometimes it flips their significance. So something that you assume to be at risk, cause of risk, is not associated with that risk, because there's actually two things. And if you don't have the second, the first doesn't do it on its own. So there may be two different markers in your genome that are needed, and that changes the whole profile.
Yes.
So that presents a challenge. So that's why you want a professional to give you the interpretation, not an online report, so to speak.
Yes. And that doesn't mean the companies are bad, but it needs to be explained. And even, we have a lot of discussion at the hospital about, what is how to explain this to patients, as we roll it out. What's the right way? What's the way to not worry somebody too much, but let them know when there's an issue, and when is that? And, it's
well, the whole concept of risk is very difficult to explain.
Yes.
If you have a 20% risk versus a 40% risk, are you actually going to do anything very different? And it depends on what is producing the risk.
Sure. Yeah, absolutely. And if it's explained in the right way, I like to think that, and this is one of the things that got us this far at the hospital, was we're very open with the patients, and we let them know what the risk is, what it's not, and give it to them in a way that they can consume. We build a lot of trust with the patients, and with something like cancer in the past, it's a lot of very intense emotional type things, but I mean, there is a way to do it. I'm feeling pretty good when we roll this out, because I feel like we've dealt with it on the most difficult end. And if we're focused on doing the right thing and explaining in the right way, maybe we can, be the best at even that part.
Yeah, but again, and this is what I tell people they saw with COVID. You're actually, the part of the challenge with COVID, I think, is people were actually seeing the scientific process as it occurred.
Yep.
Which is why the information kept changing, because you needed data and you needed to process the data. Not all the data was good. The theories changed, and then you entered into other realms beyond the science that had impact, that further complicated it. But just the underlying science is going to evolve.
Yeah. No, it's absolutely going to evolve. And a lot of what we experienced in COVID, to me, was like an ethics of how you present the information to the public debate, where people, didn't really know that. They're like, this is a hard line, answer. Do you remember Jocelyn? She had her wedding. It was a all vaccinated wedding in April of 2021. And this is when, in March, they were on TV saying, if you get the vaccine, you can't get COVID. If you get the vaccine, you can't spread COVID. That was the messaging that was going out. So her fully vaccinated wedding, about, a quarter of the people, half the people got COVID, a good amount. And she sequenced it and saw it was the Delta variant, breaking through. But that information didn't get to the public until, July or August. And, it is an ethical debate on what people should know, how much information, how you give that information publicly. And unfortunately, it turned very political as well. You had political debates out there, and we were talking about boxes yesterday. You're in this box or this box. It's like, I'm not in either box, actually. I don't even think like that, but this is how I feel on this situation. This is how I feel on that situation. And that's a lot of my experience of what science is. It's figuring out how to explain it in an honest way that, ultimately meets the proper amount of ethics in society, but doesn't go overboard or too little.
Yeah.
So, what do you think about kind of moving forward with everything else you've got going on with IPQ? Like, what's the plan for the next? I know you're big into strategic plans. What's your strategic plan for the next few years?
We're continuing the development of the women's health program, and we're working with underserved populations. We're working with African American population. We're working with the Native American population. We're even working with an Aboriginal population. Mm-hmm. We work internationally. And so part of the goal then is to use it not only to improve women's health in general, but to also start to reduce some of these disparities. Mm-hmm. And, if we can contribute to those kinds of things, I think we'll have accomplished our goal.
No, absolutely.
We don't promise to solve all the problems. Mm-hmm. We promise to help make them transparent so people can make better, more informed decisions, mm-hmm. and hopefully not confuse them, mm-hmm. but let them understand what the reality is and use that also to drive ancillary research like with the hypertension.
Yep. Well, actually, you just said informed decision, and what you just explained was what I was trying to just say too. That's exactly it.
Yeah.
That is it. How do you allow patients to make a better informed decision, and do it in the right way, and
ask the right questions.
And absolutely, and ask the right questions and look outside the box. And you always said that you look at the abstract of the question, from wide different angles, mm-hmm. To solve these complex problems. And I think by doing that, you've simplified things. We're talking about, we say it's common sense, taking a complex problem and taking a common sense approach. But, it really does in a way take your thinking outside in a different way.
Yeah.
To use critical thinking to allow that common sense approach.
Yeah. I think we call it systems thinking. We call it critical thinking, but it's understanding that what we frequently are looking at as a problem is really embedded in a much bigger system, mm-hmm. and we have to know how much of that system is it going to impact either the solution we try to put in place or what is the source of the problem itself.
And just to go back to the AI, because it's such a hot topic, you have the experience of, you said while you were with Amico, you were applying AI. You're at a lot of the AI conferences as well, and you see the booths and those type of things. Where do you think we are right now with AI, not just in health, but as a society?
I think we can differentiate AI maybe a little differently than we tend to, because we talk about AI in healthcare. Mm-hmm. There are certain aspects, AI, as we're talking about it in general, because it's a broad field, can be very useful. But it also can be very much abused. And that's what concerns me. It's abused because people get very excited. It's a new technology. They would like it to solve very complicated problems, but a lot of them don't know what the question is that they really wanted to solve, and that's where the challenge is. It's proven to be very useful in operational issues. streamlining certain processes, things where correlation can be very appropriate. But I find it still a challenge to see that it's addressing the more complicated problems. Mm-hmm. And I'm torn between whether it can going forward, or what we're encountering is, with all the hype, people are using it inappropriately or for inappropriate things, applying it out of excitement, mm-hmm. and sort of diverting us from going down the path that will give us the most success.
Interesting. That's
the concern I have.
What's the difference between AI and like quantum computing?
Oh, well, quantum computing is it's sort of like it's a mechanism that you use to allow much broader AI to be applied, bigger computers, faster computers. Quantum computing is really more of the technology that you can use
to create the AI.
You do the AI using quantum computing.
I see. And you worked with quantum computing, at least on the idea of it, for a long time, right?
Yeah.
What was the company you were working with over COVID?
Quantum Computing Inc.
I see. What do they do?
They're doing a simulation using algorithms that will be applicable to a quantum computing environment. Right now, they've moved more into the hardware side, mm-hmm. from the software side. But at the time, we were taking advantage of the fact that we could build very, very large parameter spaces.
Gotcha. So quantum computing is the computing that the AI uses to spit out the answer?
AI can use it, yeah.
AI can use it. What do you think the potential with the quantum computing is?
Again, right now, we're still challenged with building the real quantum computers. It still has to be driven by what are the problems we need to solve. Mm-hmm. It works very well in security areas. In healthcare, it still needs to be defined how it's going to be applied. We actually were looking during COVID at using it to build very complex systems to try to optimize vaccine distribution. Mm-hmm. We didn't get to do it, but it was the kind of thing where we felt if we built a system with high enough complexity, we could almost use the quantum computer to dial in changes and get very rapid feedback as to how to change distribution routes, where things were needed, and shift things in a very dynamic manner.
Gotcha. So overall, with the AI and quantum computing, am I right to say you're not sold on it being what a lot of people think it is today?
I think a lot of people, the joke I always tell people is I'm afraid that some people who are applying AI don't know how to spell it. Okay?
Yeah.
And that, it's accessible, mm-hmm. and people should play with it. But I don't know that they recognize what it can and can't do, mm-hmm. adequately.
Yeah.
I think it's still in what the Gartner calls the hype phase.
The hype phase. Have you ever seen the movie Idiocracy?
No.
I just had a thought. I wonder if we're programming this AI to be dumb, and I forget how far in the future that show was or that movie was, but if we became like these dumber beings because of programming the AI wrong to think for us.
I see some of that happening.
Yeah. I mean, ChatGPT, I enjoy it. It's very useful in many ways, but I'll set it up with questions because I feel like I know part of its bias. And
when we could spend another hour just on that, but when you become dependent on prompts, mm-hmm. then by definition you're biasing what you're going to get out. whether you recognize that or not.
Right. Yeah. So it's a big blind spot in the AI per se.
Yeah.
Yeah. That's what I found. I had a friend actually did a podcast with him that barred it said that he should be put to death, which is kind of scary for him. He's like,
Yeah.
Based on, and it made up something that he said that he never said. I used a quote from him. And it's pretty, that's, if people believe that that's, right one. I mean, I love it. Don't get me wrong. It's fun. I use it.
Again,
but
I strongly believe it has very powerful, appropriate applications.
Mm-hmm.
The challenge is people go beyond those boundaries and don't understand that they've gone beyond what its real capabilities are.
Yeah, absolutely. It can do everything. It can solve every problem, but...
Well, and I fear that a lot of society is looking for a lot of things that for technology to address.
Mm-hmm.
And, the other thing I always tell people, technology is the instantiation of a solution to yesterday's problem and doesn't know what today's problem is.
That's really good. That's really good. Anything else, Michael? I guess we can leave it with that quote, but you got anything else in closing that you'd like to say?
No, I think that one sort of sums it up.
All right. Michael Liebman, thanks so much for coming on. It's always a pleasure talking to you.
Thank you again.
All right.